Let's talk about buffer systems in the body. So, most of the internal processes which happen in living organisms are supposed to remain within a closed pH range of 7, meaning it's supposed to be mostly neutral. We can say from 6 to 8, like that, an average of 7. So, buffers are substances that resist change in pH when small amounts of acid are added to these buffers. For example, most of these buffers they are made, of course, from weak acids with their conjugate base. This is a weak acid. When we remove this hydrogen, it's going to become F minus. So buffers simply differ from each other by one. Let me say conjugates of each other differ by one hydrogen from each other. So the one which has got more hydrogens is the acid, and then the one with less is the base. Now, I want to maintain this so that it won't have that charge. Most are going to see these buffers reacted with group 1 elements, and so the charge will not be there. This is still the base, this is acid. The base is going to be one hydrogen less than the acid. So buffers are very important in certain biological molecules because they are very sensitive to change in pH. So you don't want the pH to change very much significantly. For example, the salivary amylase works well at the pH of which is slightly basic, meaning that if I get to increase the pH, it becomes acidic like that, or let me say reduce the pH, it becomes acidic. That is going to affect the enzymes. That is why buffers are important to maintain this pH, right? So what is going to happen is that buffers are able to accept hydrogen ions or donate hydrogen ions. Just make sure that they operate in this manner. So let's say we have got this buffer system as we are able to see. So if I add a base to this system, what is going to happen? The characteristic of a base is the hydroxide ion. So what is going to happen is that this hydroxide is able to grab this hydrogen and we are going to be able to form water in the process, and then we are going to have more of F minus. If we react this buffer with an acid, this acid will be able to react with this F minus and we are going to be able to form more of HF, right? So this is how buffers are going to be able to operate. So the organism's first line of defense against changing internal pH is going to be provided by these buffers. So buffers are able to include proteins, organic substances, and inorganic salts which can resist change in pH. So also nucleotides such as ATP as well as low molecular weight metabolites are going to contribute to the buffering system of the cytoplasm. Buffers are biologically important because they are going to maintain pH such that when pH becomes more acidic or more basic, they are going to be able to work on it to maintain the normal pH range. Otherwise, it can be harmful if the pH just gets to differ more acidic or more basic. So biochemical reactions are sensitive to change in pH. That is why we have got these buffer systems. It may prevent the working of some organs if the change in pH is that much, right? So these buffer systems are important in living organisms in maintaining a constant internal environment, and that is called homeostasis. Small molecules such as bicarbonates and phosphates provide buffering capacity as other molecules are going to do the same. Molecules such as hemoglobin and just other proteins, they are also involved in buffering. So there are basically three main types of buffer systems which are of physiological importance. Physiological meaning normal. If you go out of physiological, that is going to be pathological. And we have got the carbonic acid bicarbonate buffer system, the phosphate buffer system, and the protein buffer system. Starting with the carbonic acid bicarbonate buffer system, we have got carbonic acid, which is H2CO3, and bicarbonate, which is the conjugate as base of this acid. We are going to remove one hydrogen, we are going to have HCO3 minus. So this is the bicarbonate or hydrogen carbonate. So what happens is when I react a base with this buffer system, a base has got hydroxide. What is going to happen? The hydroxide is going to be able to take one hydrogen from the acid, and then we are going to form this, right? And then if I react this buffer system with an hydrogen, what is going to happen is that this hydrogen means I'm reacting with an acid is going to be able to react with this base, forming more of this. So that is what is going to be able to happen, right? So under normal circumstances, there is more bicarbonate present than carbonic acid. That is because normal metabolism in the body is going to produce more acid than base according to the needs that the body is in. So the bicarbonate is going to be regulated by the sodium, as we said earlier. You don't want to have a situation where I've got this thing which has got a negative. That is why we're going to say this attached to a sodium just to regulate the charge. So what happens is when you react this sodium bicarbonate with hydrochloric acid, because this is an acid, or let me just say if I get this buffer, the bicarbonate, the carbonic acid bicarbonate buffer, I react it with an acid. So which component is supposed to react with the acid? The component of that buffer. Basically, it should be the base, and the base is HCO3 minus, but it is attached to a sodium, right? So this is going to react with the hydrogen ion of the base, I mean of the acid, and that is going to form the carbonic acid. The chlorine coming from the acid is going to react with the sodium to form sodium chloride, which is a salt. But if I react this buffer with a base, it is the acid form of this buffer that is going to be able to react. So I'm just going to get the hydroxide. The hydroxide is going to grab one hydrogen from here, and that is going to be able to form a bicarbonate again. So this bicarbonate is going to be neutralized or is going to be regulated by this sodium that is coming from the base. So we're going to have sodium like that, and then water is also a product in this reaction. Right. So this is the carbonic acid bicarbonate buffer. So this carbonic acid bicarbonate buffer is also going to disassociate the carbon the this acid is able to disassociate to form carbon dioxide and water. So because of this, we need to expel this carbon dioxide from the body. That is why this buffer system, the carbonic acid bicarbonate buffer, is related to the respiratory system because you have to remove it via breathing. Okay. So in case they asked us to mention which buffer system is related to the respiratory system, that is the carbonic acid bicarbonate buffer. Also our phosphate buffer systems. So the phosphate buffer systems mostly operates in internal fluids of cells. So that is internal fluids of cells inside the cells. So it's going to consist of dihydrogen phosphate, that is two hydrogens and then a phosphate. A phosphate is supposed to be PO3 minus, but I've reacted with two hydrogens. The charge is going to increase by 2. So from 3 to 2 to -1, like that. So this is one component of this buffer. Another component is the hydrogen phosphate, which is HPO4. So this is going to have a charge of -3. Now to just regulate these charges, this one has got a charge of -1. I'm going to react it to one sodium to regulate this charge. This one I'm going to react it with two sodiums. Okay, so this is sodium dihydrogen phosphate. This is disodium hydrogen phosphate. These are the components of this phosphate buffer system. So these are also going to be able to prevent any change in pH. So when we get to add an acid, what is going to happen? If I add an acid, it is the basic component of this buffer that is supposed to react. And the basic component has got an hydrogen lower than the acidic, meaning that this is the basic. This one is able to react, so meaning I'm going to have this HPO4. Let's say I've reacted it with a strong acid like hydrochloric acid. So the hydrogen here, what is going to happen is going to be attracted to the bike to this hydrogen phosphate, and what we are going to be able to form is dihydrogen phosphate like that. The chlorine is able to react with the sodium because we have got sodiums, two sodiums here. So one sodium is going to react with this chlorine to form sodium chloride, and then the other sodium that has remained is able to react with this compound in front here to regulate it so that the charge will no longer be there. Meaning if I reacted this with a base, what is going to actually react is this one, which is the acid. Okay. So this is basically what gets to happen in this buffer. So when sodium hydrogen phosphate, disodium hydrogen phosphate encounters a strong acid, as we've already explained this. So the reaction that is going to happen. And then when it is a strong base, which this buffer is going to experience, it is a sodium dihydrogen phosphate, which is an acid which is going to be able to react with the sodium hydroxide. Remember, the buffer system is made up of weak acid and weak base. We also have protein buffers in blood and cells. So amino acids that make up proteins can be positively or negatively charged with carboxyl groups. So you know that amino acids have got a carbon bonded to an R group and hydrogen. We have got a carboxyl group. It's supposed to have an hydrogen here and then an amine group like that. Now, most of the times we're going to see this hydrogen being attached to the amine, and so this is going to be positively charged, this is going to be negatively charged at physiological pH, and so this is going to be called the deuterium. So what happens is when I get to add an acid to this buffer, that acid, it has got hydrogen ion. This hydrogen ion is going to interact with this part, and that is going to attach here, so meaning that the amino acid is going to remain positively charged. If I react this with a base which is hydroxide, this base is going to grab one hydrogen from here. We're going to remain with only two hydrogens. So meaning that this amino acid is going to remain negatively charged. So this is what gets to happen. So these charged regions can bind hydrogen or hydroxyl. These are the charged regions, this region and this region. And that is going to make proteins to act as plasma buffers, right? So most of the buffering capacity of blood and cells within it comes from protein. That is for blood and cells. Most of the buffering is going to come from proteins. We're going to see there are some other buffering systems that are able to find in blood, but mostly these are the ones we're going to see. But for the whole body, the most abundant buffers that we have are the carbonate buffers. Red blood cells do contain hemoglobin, which carries oxygen. Of course, hemoglobin also is able to carry carbon dioxide from the tissue where it is produced, taking it to where it has to be removed. That is why we said on the buffers of that is the carbonic acid and bicarbonate, we said there is carbon dioxide produced, which is going to be transported via the respiratory system. So basically, the carbon dioxide is able to attach to the hemoglobin, just like oxygen does attach. Now, what happens is because we saw that the carbonic acid can be broken down to carbon dioxide and water, this carbon dioxide and water can still react to form carbonic acid like that. This carbonic acid can further break to form a bicarbonate and hydrogen ion like that. So conversion of carbon dioxide to the carbonic by what does this work of conversion from carbon dioxide to carbonic acid is done by carbonic anhydrase enzyme that is going to form the bicarbonate which you're able to see here and also the hydrogen ion. So hemoglobin is able to bind the hydrogen ion that has been formed by the action of carbonic anhydrase. But this ability is reduced once oxygen becomes unbound to hemoglobin. Okay, so the binding of hydrogen to hemoglobin is going to be reduced when oxygen is not bound to hemoglobin, and that is going to mean that hemoglobin, when it does, of course, enter the cells, because in the cells we are getting to produce carbon dioxide there, it will not be able to pick up these hydrogen ions, meaning there will be accumulations like that. Okay, so this is a table that summarizes where we're going to be able to find these buffer systems. Outside the cell, that is in the extracellular fluid, we're going to find the major buffer is the bicarbonate buffer system. And then we also have minor ones like the proteins which are outside the cell and the phosphate buffer system. In the blood, the major buffer system we're going to be able to find bicarbonate buffer system and also the hemoglobin buffer system. Okay. We saw, of course, in the blood, if we talk about these buffer systems, we're not excluding the protein buffer system because that is the major in the blood plasma like that. And then the minor ones we have got the plasma proteins because just inside the blood there we have got plasma, and then phosphate buffer systems. And then inside the cell, we already mentioned this. Inside the cell, I've got phosphate buffers and also protein buffers because we've got a lot of proteins inside the cell. And then in the urine, we have got ammonia buffers and phosphate buffers. So what of if they told us to comment on the, let's say, GIT? What kind of buffer might be there? GIT, that is outside the cell, extracellular. So what do we want to expect? Want to expect bicarbonate buffers. So bicarbonate buffers are mostly out, let's say, abundant. We can see they are appearing like almost everywhere as major buffers. So they are major buffers of the whole body.